My name is László Boros. I'm a professor of pediatrics at the University of California, Los Angeles, UCLA. I teach medicinal biochemistry, and I do teach deuteronomics, which is the deuterium discrimination process throughout nature, or processes that take care of nature. Those are biological systems and physical systems that actually discriminate between hydrogen and deuterium, which are two isotopes of the first element of the periodic table, hydrogen. And because of the mass effect, because of the kinetic isotope effect, it seems that this type of knowledge or this type of biochemistry interpretation of the kinetic isotope effects of deuterium have great clinical applications. And me as a UCLA professor I'm working on establishing these clinical scenarios in the translational clinical arena of medications to treat various human conditions that include cancer, bronchopulmonary dysplasia, which is a lung disorder in stillborn babies, obesity, diabetes, practically you name it. It seems that deuterium depletion has almost a significant important role in almost all aspects of medicine, including treating chronic diseases, neurodegenerative diseases, cellular degenerative diseases, and so on. In 2002, there was a colleague of mine in Los Angeles who actually introduced me to Gábor Somlyai, who was working on deuterium depletion. At UCLA, I was running a laboratory where we actually phenotyped, we studied cells in culture in response to various agents, including drugs, and we used the labeled glucose molecule to follow its fate in cells, and we could do this very precisely using mass spectrometers. And using this technology, advanced biochemistry phenotyping technology, we looked at deuterium depleted water under an arrangement with Dr. Somlyai, and to our best and biggest surprise, there was a dose-dependent effect of deuterium depletion on cellular substrate oxidation, and we presented this in 2006 at the metabolomics meeting in Boston at the Harvard University. Then it was a few years, then the next clinical problem showed up from the National Cancer Institute of the United States, where they actually presented a patient who had one of the mutations of the metabolic enzymes that recycles deuterium depleted matrix water in mitochondria into the tricarboxylic acid or St-Györgyi-Krebs cycle. And sure enough, there was an important link that we could position deuterium depletion and deuterium depleted water in the mitochondrial functions, and we now know that actually that's one of the most fundamental processes in living cells. It's really not that how much you understand, this is a fact, it's undisputable. Practically from fatty acids, from sugars, from carbohydrates, what you do is you harbor your protons or hydrogen, and if those are deuterium-loaded substrates, then your mitochondria is not able to produce the deuterium-depleted water, and the result of that is disease, including cancer, diabetes, obesity. We call it molecular crowding or metabolite crowding because there's these little nanomotors in your mitochondria which actually spin. Physically, you have to consider these like little nanomotors. That's how they call them. They are driven by hydrogen. And if they encounter deuterium, actually they suffer a structural damage. They stutter, they stop, they break their proteins. And there's also a new phenomenon called the quantum destabilization of water, which is proton related. This process is practically making protons available for becoming more of a wave kind of a physical phenomenon, so they lose their masses. It seems that between hydrophobic surfaces, protons change their phase, but they go from a mass or physical form more of a wave kind of energy state. We call it 0-point energy. And it seems that this is also part of mitochondrial functions, because in mitochondria there are membranes, hydrophobic membranes that can actually do the same with protons. This is the study from University of California, San Diego. And actually, we now link these inorganic physics phenomenon with mitochondrial functions, and this process of this quantum destabilization of protons is compromised by deuterium. So practically, this process being so crucial in mitochondria, but being compromised by deuterium, deuterium depletion is the only way to protect mitochondria from deuterium-related damage. And one of the ways of depleting deuterium in our system is through deuterium-depleted water. There are other ways, obviously, through nutrition. And we are exploring these scenarios, these interacting biochemistry-related principles to use them in medicine. Now we actually understand the basic physics and biochemistry of hydrogen-deuterium interactions. In 2015, with Gábor Somlyai, we published a paper with collaborators from the Johns Hopkins University, University of South Florida, and from the University of Arizona, where we actually linked mitochondrial functions with deuterium depletion. That actually pushed the field towards more mitochondrial studies in various deuterium environments, and those results are now discussed. And the new phenomenon that I presented is this quantum destabilization of protons or water, which is also part of the deuterium-related kind of developments, and we hope that this meeting will also bring this new type of information to the focus of these great investigators. So practically, it's moving towards medicine. It's moving towards clinical medicine. It's moving towards translational medicine, and this is the best forum to discuss these developments and see how the future will turn out for us.